Hi everyone and welcome to the internet. What we're going to be doing today is having a look at mutations. So we're going to know what they are and what different types they are. What I'd like you to do first of all then is have a go at our 3 starter questions. So answer these in your books. Okay, let's mark those then. Question 1, the dominant are shown by a capital letter. Mark for that one. For question 2, both alleles are the recessive alleles. Tick for that one. And then for number 3, if you got the gametes for the mother as the 2 X's in individual circles, 1 tick. If you got the father's gametes as 1 X and 1 Y, tick for that one. If you got the 4 genotypes, so XX, XX, XY and XY, tick for that one. If you got the 4 phenotypes, so 2 female as the XXs and the 2 males as the XYs, tick for that one. And then if you wrote down the percentage as 50% chance of male or 50% chance of female, give yourself a tick for that one. So give yourself then a mark out of 7 and write that in your books. First thing I'd like you to do then is write down what you think a mutation is. When we talk about mutations in biology, what we're referring to is a change in the base sequence in the DNA. So if you got that, give it a tick. If you didn't get that phrasing, make sure you've written that down as the textbook definition, if you like, for it. So a change in the base sequence in the DNA. See if you can write me down a list of anything that you think causes mutations. Just 1 minute to have a go. When it comes to mutations, obviously you can mark any of these you got right and make sure you've got these written down if you didn't have them. The first and most important point we need to remember is that mutations can occur spontaneously. So throughout your life, then there will be mutations that occur within your DNA. But we also see that there are certain factors that increase the chance of a mutation occurring. And the two key ones that we really need to remember here, firstly, ionizing radiation, and a really good example of that is UV, which comes from the sun. And the second one are exposure to certain chemicals. So things like ethanol, things like benzene, these will all increase the chance of mutation occurring. Have a go answering this then. Why would sunbathing increase your risk of skin cancer? Hopefully we managed to put together the idea that if you're sunbathing, then you're increasing the exposure of your skin to that ultraviolet radiation, the ionizing radiation. Now, what happens is when your skin cells are exposed to that ultraviolet radiation, then it can lead to damage in the DNA of your skin cells. And that damage can lead to skin cancer. And that's why it's so important that when you are outside, then you are wearing sun cream of an appropriate sun protection factor. Because anytime you're actually burning yourself, then what you're doing is that's your body's way of saying you've caused damage. It's a bit late to do anything about it, hence why we use the prevention of the sun creams. So whenever you're going out in the really nice weather we've had of late, then sun cream is important. What we've discussed in some of our earlier lessons on genetics is this idea of variation. So we've talked about the fact that we do have variation within a species. Now, the reason that we've got that variation is because we've got a different version of an allele. And that's arisen from a mutation that changed the DNA. So where we've got all of these different eye colours, skin colours, hair colours, everything like that, they've all come from mutations within those particular alleles, leading to these different phenotypes that we now see. But not all mutations will show the same effects. When we get a mutation, some of them will show absolutely no effect on the phenotype at all. So what we see is even if you have a mutation, it won't always present as a change in the phenotype, the characteristic we can see. It may influence the phenotype, so it might be a contributing factor, but not the overwhelming deciding factor, if you like. So it might just give a little bit of a change. Or it may overall determine the phenotype. And it's that last group there, where the mutation determines the phenotype, that we get those variations within a species. So make sure you've got those points written down. I'll leave it up for another minute and a half. Now, as I've mentioned, throughout your life there will be a mutation or two that occur within your body. But it's not a case of if you have a mutation then it's instant death. Because mutations come into three categories. We've got harmful mutations, we've got beneficial mutations, and we've got neutral mutations. And we're going to look at each of those in turn just to see what we mean by those and some examples of them. If we start off with the harmful mutations first of all, which are probably the ones that your brain jumps to straight away, then the first really good example of a harmful mutation are the ones that cause cancer. And where we talk about cancer, this is basically a condition in your body where your own body cells are growing and dividing in an uncontrolled manner, which is why it's so tricky to treat, because any of the drugs that we're using that target the cancer cells, well, they're just normal body cells. That's why we've got so many side effects with cancer treatments there. Second harmful mutation are the ones that cause abnormal channel protein formation. Now, channel proteins, if we think back to our earlier biology work, are these kind of little channels that go through the cell membrane. So they're a specific shape to allow certain molecules to cross from one side to the other. And if that channel protein isn't produced normally, so we make a different shape or we don't make enough of them, for example, then we get problems in that process. And a good example of a condition that is caused by this abnormal channel protein production is cystic fibrosis. The third one there is that we can actually cause different shape protein molecules to be made. And a good example of this is sickle cell anemia. So your red blood cells are normally that biconcave disc shape if we've not got sickle cell. But if you do have sickle cell anemia, they change from that disc shape into more of a crescent moon shape, so shaped like a sickle. And that means that they're not going to be as efficient at transporting oxygen around your body. So make sure you've got those examples written in your book. I'll leave them up for about another 40 seconds. When we talk about neutral mutations, these are ones that have absolutely no bearing on your chance of existing and surviving at all. So good examples of this is your ability to roll your tongue and whether you've got 3 earlobes or whether they're attached to the side of your head. It really makes no difference whether you can roll your tongue or not. It's not a case of if you can roll your tongue, you're definitely going to survive to the age of 90, whereas those who can't roll their tongue, they're doomed by the age of 60. It's just something that we can either do or can't do, but it doesn't impact on survival. The third type of mutation we've got are those that are beneficial. Now, if we consider one example that's not in humans first of all, so something that occurs in bacteria, we've got that mutation for antibiotic resistance. So that means that bacteria now won't be killed by the antibiotics that we take to get rid of them. So it's beneficial to the bacteria, not so much for us. Second one is the FOXP2 gene, and this was a mutation that allowed us to be able to speak. So if we hadn't had the mutation in the FOXP2 gene, we would not be speaking as we do today. And the third one on there is the skin colour mutation. So hopefully we know that if we track back our human history, then we originate from a small number of breeding pairs that originated on the continent of Africa. Now, if you are in Africa, it's actually beneficial to have much darker skin because that protects you from absorbing too much ultraviolet from the sun and therefore reduces your risks of skin cancer. When we moved to lovely climates like the UK and obviously those places away from the equator, then the amount of ultraviolet in the sun was decreased. So the intensity of the sun's rays as we move further from the equator does decrease. And actually having darker skin isn't an advantage there. So we had this mutation within the genes that control skin colour that led to the lighter skin tones, and that means that when we're living in countries like England where for some years it's pretty much cloudy and rainy almost all the time, then we can still absorb enough of the sun's rays to make enough vitamin D to stop us getting conditions like rickets, for example. So mutations in skin colour are beneficial as well because it's allowed us to live successfully in a range of different countries around the world. Have a go at answering the question on the screen in your books then. Let's double check to make sure we got the key points. So hopefully we know that people with darker skin then have reduced amount of UV absorbed by the skin cells. And it's more important that this is true of the skin cells deeper within the actual body, because those are the ones that are more susceptible to damage. Because they're absorbing less of that UV with those delicate cells, then they're less susceptible to damage, which means you reduce the risk of skin cancer. If we have a mutation to give lighter skin, then in those countries that have lower intensities of sunlight, then the skin can still absorb enough to make vitamin D, and people with the darker skin tones in countries that are quite dark and dingy, then they can actually struggle to absorb enough to make the right amount of vitamin D. So some people may actually have to take supplements of vitamin D to actually stave off those conditions that require vitamin D to avoid. Before we leap into what's actually going to happen, then see if we can remember the process by which we make a protein. So protein synthesis. See if you can explain how a protein is made. So you get your first mark if you said the DNA for the gene unzips. Second mark, 3 RNA bases in the nucleus pair with the complementary base pairs on the DNA. Third mark for saying the mRNA strand leaves the nucleus and travels to a ribosome. Fourth mark, the ribosome reads it in triplet codons or sequences of 3 bases. Next mark is for saying that each triplet corresponds to a tRNA molecule which is attached to amino acid. Next mark is for saying the tRNA brings the amino acid in and they join together to make a peptide chain. And the final mark for saying that it then folds to make the protein. Give yourself a mark out of 7 based on that. And obviously if you want to write this down as a reminder, hit pause on the video and just jot those points down. The big issue with changing these triplets and therefore changing the amino acids we code for is that when we change amino acids, it can impact on the way that the protein actually folds up. So it changes the 3-dimensional picture or structure of the protein. And that means we could actually create a completely different protein. And that's obviously problematic because proteins can be things like enzymes, and the shape is very important to those. So let's see if you can tell me why then. Have a go at answering this question in your books. If a mutation occurs in an enzyme, then as we've said, the three-dimensional structure of the protein is changed. Now that means that the shape of the active site could change, and that means it's no longer complementary to the substrate. So the substrate can't bind to the active site because they're different shapes to one another. The enzyme can't work. Last thing for today then is to answer the plenary questions. So 3 questions on the screen, have a go at answering those in your books. I'll leave them up for a couple of minutes today, but as always, if you need any longer, hit pause and take as long as you need.